Okay, guys, we've finally made it. We're on the final straight. Okay, we're going to talk about OSNAS rice. So I mentioned this last year in term one, and then we looked at some news reports recently on it as well. I handed out those news issues in class. So I just want to go over it again in a little bit more detail. So OSNAS rice. Why do we need OSNAS rice? Well, like we said, there's an iron deficiency globally. There's people that are not getting enough iron in their system, and an iron deficiency can cause a bunch of consequences. Okay, reduced physical and cognitive development in children, increased child mortality, and reduced work productivity in adults. Iron helps your body to create healthy red blood cells, and it helps you carry more oxygen in those blood cells. When your body doesn't absorb enough iron, you don't get enough oxygen and you feel tired and weak. So you may have seen this. People have iron deficiencies naturally. They can't really take iron in from their food. You can maybe get iron tablets. And people who have an iron deficiency can be quite lethargic and tired. So that can be a deficiency that you have naturally in your body no matter what you eat. But a lot of people in the world just don't have enough iron in their diet. So that's why we have OSNAS rice. Okay, why OSNAS rice? Well, rice is the primary source of food for billions of people in developing countries, so it's grown in these areas where deficiencies are high. Yet, commonly consumed polished grain contains insufficient levels of key micronutrients such as iron, the chemical symbol is Fe, zinc, and the chemical symbol is Zn, and vitamin A to meet daily dietary requirements. So, rice is the primary food source, but it doesn't have all the nutrients, mainly iron, that people need. So, what has biotech done? Well, some experts have estimated that a rice-based diet should contain 14.5 micrograms of iron in the endosperm. So the endosperm is the large section of the rice here. Okay, so the main part of this endosperm, the part of the rice grain, should have 14.5 micrograms of iron, minimum. The main constitute of polished grain, but breeding programs have failed to achieve even half of that value. Transgenic efforts to increase the iron concentration of rice endosperm include expression of the ferritin genes, nicotianamine synthase genes. Thank God they've used a smaller term called NAS genes, NES, OSNAS. Okay, that's where the NAS comes from. So if they look at this part of the gene, essentially what they're saying is, if they put this, say this gene is called NAS gene, when they express this in the rice, so they turn it on or elevate it, then you get a higher production of iron. That's essentially what they're saying, okay? So we're going to go do a little bit of jargon, but that's basically what they're saying. When they turn this gene on, in the rice, there's a higher production of iron in the endosperm. Perfect. So how did they do this? How did they prove that these NAS genes were expressing more iron? Well, 3 populations of rice were generated to express OSNAS1, OSNAS2, and OSNAS3. So there was 3 NAS genes, and they wanted to see which one produced the most iron. So they switched on OSNAS1 and put it into one rice plant. They switched on OSNAS2, put it into a second rice plant, and they switched on OSNAS3 and put it into a third rice plant. And what they did is they grew the 3 different types, OSNAS1, OSNAS2, OSNAS3, and they had to look at the endosperm, okay, because remember that's what they said, that's where the iron was being collected. So how did they do that? How did they know how much iron was then in these rice plants? Well, they did something that sounds like a transformer. They used a Synchrotron X-ray fluorescence spectrometry. Cool. So essentially what they did is they just put an X-ray through the rice grain into the endosperm and to see which one produced the most iron, essentially. Okay, so I'll just read that. They X-rayed the endosperm of the rice. The OSNAS genes, particularly OSNAS2, showed enormous potential for iron and zinc biofortification. So the word biofortification, we think of fortifications as things like castles, or if you create more fortifications, then you're kind of strengthening an area. So you maybe, that's where you use the term castle, because you fortify your position in a castle. So biofortification is you're improving the amount of a biological element, in this case, iron. So this biofortification is a word they may use. Remember when we said in the HSE questions, you know this stuff, but they might use something you're like, oh man, what does that mean? Biofortification, so they're improving the amount of iron. They're fortifying the rice with iron. The results demonstrate that the rice cultivators overexpressing single rice OSNAS genes could provide a sustainable and genetically simple solution to iron and zinc deficiency disorders affecting billions of people throughout the world. Okay, so let's just explain that a bit more simply. So after they x-rayed the endosperms, so the main part of the rice, they found that the rice that had the OSNAS2 gene produced the highest amounts of iron and zinc. Therefore, if you can turn that OSNAS2 gene on and produce that rice crop, the OSNAS crop, then you're going to increase the amount of iron and zinc available in the rice crop and therefore reduce the deficiencies that people have in developing nations. Okay, so I'd maybe review that again, but we know essentially OSNAS genes produce more iron in the endosperm. OSNAS2 had the highest proportion, so this one here, had the highest proportion of iron and zinc. How do we know that? Well, we x-rayed the endosperm and it showed us that OSNAS2 had the highest levels. If you want to remember this transformer name, Synchrotron X-ray fluorescence spectrometry, go to town. But essentially the x-rayed endosperm and proved that there was a high percentage of iron, particularly in the OSNAS2 plant. And why is that important? Well, research is a necessary component in the development of technologies as it plays a role in regulating and testing new technologies to ensure that they are not a breach of biosecurity, human health, long-term sustainability, and within social ethics. Research needs to be independently conducted in order for the results to be unbiased and thus provide a balanced evaluation of the agricultural technology. So think back to your investigating science stuff you've been doing with Chris. We're talking about making sure that these experiments are fair and unbiased, so they're not just being done because, say, Monsanto wants it to be look like it's an amazing crop so they can make loads of money. This is particularly important with advancements in genetic technology and its application in agriculture, as the direct manipulation of genetics is still new and improving field, with little knowledge of the long-term effects of genetically modified organisms on our health and the environment. Okay, so that's quite a nice little summary of why this is important, but how we need to kind of tread carefully with biotech more generally. Okay, my final bits of advice for you when you walk into your HSC or you strut in like that cow at the beginning video. The first thing you're going to do is you're going to write a cheat sheet on the back of your HSC paper, okay? I want you to, when they say you can turn around your paper, I don't want you to turn it around. I want you to write down things like can't play golf because of crazy raptor, ethylene, all your plant hormone stuff, xylem, phloem, OSNAS rice, BT cotton, EnviroPig, Aqua Advantage salmon. Okay, I want to see all these terms just spew out, vomit out onto the back of that page. Because then when you go through your paper, you've got a cheat sheet already at the back, okay? Just do a big brain dump on the back of your page with all the information that we've learned about, and then start your exam and refer to that. And that's going to settle you as well. You're going to feel a bit less anxious because you're like, oh man, I've already got my cheat sheet at the back, okay? So you're already ahead of the game. I want you to look at your plant hormones, plant anatomy, and animal digestion, the things that I didn't cover in these videos. There's things that I obviously couldn't cover in all of these videos that we have looked at in class, so please refer to your folders. All the information's in there. Attempt every single question. If your hand is not sore by the end of your exam, you've done something wrong, okay? I know it's a long time. 3 hours is a long, long time. But nobody used the full 3 hours in the HSC trial, and you have that time, okay? How many years have you been at school? 13 years maybe? 3 hours is nothing. Just sit there and put as much information down as possible. The only guarantee I can give you in that exam is if you leave it blank, you get no marks. If you put something down, you could pick up marks. Put something down. And if you really don't know the answer, please don't do anything stupid, right? Oh, F this, I don't know the answer, or this is effing stupid. You may get frustrated in the exam, I understand that, but if you write this is F student but are F and hate agriculture, they will just take away your whole exam. They won't mark the thing. So you don't want to do that. Okay, keep the frustration, go outside and shout it afterwards, but don't write it down. Your marker will just void your exam paper. Okay, and that's just a bit daft. So please do some revision. Get in touch if you need any more help. Hopefully these videos have been some bit of use to you. And yeah, good luck with the studying. I'll see you before your exam, and you'll be fine.